Hi, my name is Stephanie Serrano, and I welcome you to the first chapter of my podcast, Lymphocyte News, where I will be talking about the latest news in the world of biomedicine and immunology. Let's begin. In order to inaugurate this podcast, I have to ask you a question. Do you think it is possible that there will be a cure for HIV in the near future? One of the deadliest diseases of the century? Well, let me tell you that on October 27, an article was published in the journal Medicine Nature about a clinical trial under development for a possible cure for HIV, which combines antiretrovirals with immunotherapy, creating a therapeutic strategy that enhances the response of the immune system against the virus and decreases the number of infected cells. As is known so far, the main problem in developing a cure is due to the great ability of HIV to hide in the long term from the immune system in regions of the human body called reservoirs, which is why the treatment should be chronic in almost all people. If the drugs are stopped, the virus multiplies again and spreads throughout the human body from the reservoirs and attacks the lymphocytes, defensive cells, which eventually causes immunosuppression. Until now, numerous investigations throughout the world have focused on finding a curative treatment, that is, one that managed to definitely eliminate HIV from the human body, instead of keeping it at bay every day with drugs. For now, efforts over decades have been unsuccessful to achieve this goal in the general population. The recent study that we will talk about today invites, to say the least, optimism. A combination of antiretroviral therapy together with immunotherapy has shown very promising results in patients with HIV. The results show that this treatment scheme improves the immune system response to the virus and favors its elimination by destroying a greater percentage of infected cells. The research, in which members of the AES Research Institute in Spain have participated, consists of a phase 1 and 2 clinical trial. Participants were 55 people recently diagnosed with HIV-1 infection, divided into 4 study groups. Group 1 received the usual treatment, Group 2 received the standard treatment plus an antibody against HIV called 3BNC 117. Group 3 received the usual treatment plus the drug romidopsin that is used against lymphomas and that was used here to make it difficult for the virus to infect the cells. Finally, Group 4 was administered the 3 groups of drugs: standard therapy, the 3BNC 170 antibody, and romidopsin. The researchers examined participants' HIV viral load and the proportion of infected CD4 T cells from before treatment to 1 year after treatment began. By administering standard antiretroviral therapy early along with the HIV antibody, they found that HIV suppression was accelerated and also the elimination of infected cells, lymphocytes, compared to those who received only the usual treatment. The next part of the trial involved stopping antiretroviral treatment in 20 volunteers for 12 weeks. The authors found that 5 of the 6 volunteers who received the 3BNC 117 antibodies were infected with 3BNC sensitive virus variants, kept their viral load under control, even though they were no longer taking antiretrovirals. In addition, in one participant who received the 3 groups of drugs, the RNA of the virus was not detected in the blood throughout the follow-up time. The authors maintain that the combination of standard antiretroviral therapy applied early together with the 3BNC117 antibody enhances the elimination of HIV both in the blood and in infected cells. It is still early to consider this treatment scheme a cure against HIV. In any case, it represents a step forward in preventing the virus from remaining indefinitely in the human body, which could make it possible to dispense with antiretrovirals for life. However, it will be necessary to see what happens in the third phase of this investigation. Continuing with other news, can you imagine that we could travel to the past to save our present, like in the movies? A Cornell University article suggests something particular, that viral DNA in human genomes, embedded there from ancient infections, serve as antivirals that protect human cells against certain current viruses. Previous studies have shown that fragments of ancient viral DNA called endogenous retroviruses in the genomes of mice, chicken, cats, and sheep provide immunity against modern viruses that originate outside the body by blocking them from entering host cells. So this study was conducted with human cells in culture in the lab. It shows that the antiviral effect of endogenous retroviruses likely also exists for humans. The research is important because further inquiry could uncover a pool of natural antiviral proteins that lead to treatments without autoimmune side effects. The work reveals the possibility of a genome defense system that has not been characterized but could be quite extensive. The results show that in the human genome, we have a reservoir of proteins that have the potential to block a broad range of viruses, said Cedric Fechot, professor of molecular biology and genetics in the College of Agriculture and Life Sciences. When retroviruses introduce their RNA into a host cell, the virus hijacks the cell's transcriptional machinery to replicate itself. Typically, retroviruses infect cells that don't pass from one generation to the next, but some infect germ cells, such as an egg or a sperm, which opens the door for retroviral DNA to pass from parent to offspring and eventually become permanent fixtures in the host genome. In order for retroviruses to enter a cell, a viral envelope protein binds to a receptor on the cell's surface, much like a key to a lock. The envelope is also known as a spike protein for certain viruses, such as SARS-CoV-2. In the study, Fechot and colleagues used computational genomics to scan the human genome and catalog all the potential retroviral envelope protein coding sequences that might have retained receptor binding activity. Then, they ran more tests to detect which of these genes were active, that is, expressing retroviral envelope gene products in specific human cell types. We found clear evidence of expression, Fechot said, and many of them are expressed in the early embryo and in germ cells, and a subset are expressed in human cells upon infection. Once the researchers had identified antiviral envelope proteins expressed in different contexts, they focused on one, suppressin, because it was known to bind a receptor called ASCT2, the cellular entry point for a diverse group of viruses called type D retroviruses. Suppressin showed a high level of expression in the placenta and in very early human embryonic development. Then, they ran experiments in human placental like cells, as the placenta is a common target for viruses. The study shows how one human protein of retroviral origin blocks a cell receptor that allows viral entry and infection by a broad range of retroviruses circulating in many non-human species. In this way, Fouchot said, ancient retroviruses integrated into human genome provide mechanisms for protecting the developing embryo against infection by related viruses. Future work will explore the antiviral activity of other envelope-derived proteins encoded in the human genome, he said. But for now, we know that ancient retroviruses could save us from present viruses. We have come to the end of this chapter, and I'm very thankful if you listened to the end. And see you next week.